Okay. Let's pick up where we left off. Does that look familiar? How about that? So we just got done talking about Gerard. 1941. So, and I talked about Pearl Harbor and my FDR quote. So we're in the middle of Beetle and Tatum. What were the results of Beetle and Tatum? Okay, nobody knows. So that basically means we're in the middle of Beetle and Tatum, right? So so basically Beetle and Tatum was the experiment. It's actually a pretty good one, whereby we connected Gerard. So Gerard was basically looking at mutations, right? He was looking at alkaptonuria. And one of the things that Beetle and Tatum figured out was that we could actually follow mutation ourselves, not just natural variation, right? So we don't have to wait around for like a purple color or a white color, because if you think about it, we're a little more limited. We're just looking for natural variation. Not only that, but we don't learn a whole lot about natural variation, because there's a lot of things we don't see and we can't follow, like diseases, for instance, right? You don't necessarily have the ability to follow alkaptonuria because it's a metabolic thing. It's not a visible thing. So what Beetle and Tatum figured out is that you can make mutations in genes, and basically mutations will follow the same rules as normal variation. Which is good, because what that means then is we can go in and we can blow something up, which we're really good at, and then we can learn about what it does by seeing how that function is compromised. Okay. So, and that's kind of, so that would allow us to be able to learn a lot more, especially like in a metabolic reaction where you don't see it visibly on the outside. It's something you see on the inside. And so what they did was they essentially worked with Neurospora crassa, which is a fancy name for bread mold. And so what they did is they did a mutagenesis screen on it. So they basically took Neurospora, they blasted it with X-rays, right? So what they're doing is a mutation. So they're creating lots of mutations that are randomly scattered throughout the genome. That's what happens when you kind of get mutated, right? So you don't just mutate a gene, you mutate all of them, right? So if you submit yourself to radiation, you're going to get mutated, but it's going to be random mutation across all your genes. The idea is hopefully one of those genes is one you're interested in. The rest of them you don't care about. So you want to basically screen those out and sort of eliminate those. And so that kind of gets you to the next step, which is looking for nutritional mutants is what they did. So this is kind of a selection process that they actually use. And so their selection process was essentially to look for nutritional mutants. And we do the same thing now, right? So at UC Davis as an undergrad, one of the things I did was work with the mutation screen. So we essentially were looking, it was a meiosis lab. And so we were looking for mutations in the meiosis process, right? So the idea of meiosis is that there's proteins involved in pulling it all off, and we wanted to know what those proteins were. And we basically look for a scorable phenotype, right? They were looking for nutritional mutations. We, in our lab, were looking for evidence of meiosis dysregulation, right? So the look for would be non-disjunction. Right. So you'd either have too many chromosomes or not enough chromosomes. And so that was our score. So we'd look for anything that, or any fly that exhibited really high rates of non-disjunction. Because the idea was whatever's happening with that fly, there's a foul up in their normal meiosis, and we want to understand that. So they were looking for nutritional mutations. And the important thing about Neurospora is, like several organs in their same taxa, these guys can make their own amino acids. They have genes that allows them to make their own amino acids, which is kind of cool, right? We have to consume ours, they make theirs. And so what that means is if we were to take neurospora bread mold and we were plated on what's called minimal media, this is like bare bones nutrition. Only healthy individuals can grow on this. So if all of your genes are working, all of your amino acid, your nutritional genes are working properly, then you'll be able to live on minimal media. If you have a mutation in a gene, you will not be able to live on minimal media because you have an essential gene that's just not working by blasting it with x-rays. So what they really wanted to do then is they wanted to basically take a look at this, and they wanted to understand a little bit of something about the biochemical pathway specifically for one amino acid, and that's arginine. They knew there were different things involved in the production of arginine. You don't just go straight to arginine. There were several different chemicals involved in, like ornithine or arginosuccinate. They weren't quite sure like the order of them, but they knew that they were involved. So they knew that there was a little bit going on there. And so they wanted to understand this. What they did is they basically created a screen to generate arginine mutations. So their selection wasn't just for nutritional mutations, but specifically for mutants in arginine production. Now remember, when you do a mutation analysis, it's going after all the genes. So when you score for your particular phenotype, in this case, the inability to make arginine, then you also have to understand that there's a whole host of everything else is also mutated. So you're like picking your needle out of the haystack, and then the idea is you just ignore the rest. Beetle data basically are credited with really pinning down for us sort of a modification of Gerard. And they basically kind of came to the one gene, one enzyme hypothesis. That is to say that a gene is responsible for encoding an enzyme. And that's kind of where that all started. So let's go ahead and take a look at their actual results. Here's basically what they did. So they took wild type neurospora, that's normal neurospora. They basically blasted it with x-rays. And the important thing is that when you blast something with x-rays, this is true for all mutagenesis, right? We have two major types of mutagenesis that we do in the lab when we're trying to generate mutations to study and follow. One is with x-rays or radiation, right? Cesium chloride, for instance, is a common source that we use. The other one is with chemicals, right? Notice from the previous list, our two main mutational problems are radiation and chemicals. Those are the two things that we use to generate mutations above background. Now, what's happening is when you radiate these guys, I mean, you're just beating the devil out of them. I mean, so the cells are accumulating all these mutations. I mean, they're like on their last leg, right? And so the idea with mutation, it's a little bit of an art, right? You want to basically blast your cells so they accumulate lots of mutations for you. And you want to push them like right on the edge of viability. But you don't want them to go over because then they'll be dead, right? Because you want them to live and you want them to carry the mutations that you want to study. So in order to do that, it's kind of like what we do to chemo patients, right? We blast their cells with these very cytotoxic. They are not in good shape at all. But what do? We allow them a chance to recover, right? So that we can do it again. As long as we need to in order to get the upper hand on the cancer, right? The same thing is true here. So the first thing we do then after we blast them with x-rays is we put them on rich growth medium. So this is basically the lap of luxury. The Thanksgiving banquet. I mean, you want 12 courses, you have 15 courses if you want, right? You want to eat cake, eat it, right? You want, I mean, just think of it. We'll get it for you. You want a hot tub? We'll make it happen for you. You want a yacht, a mansion? We'll make it happen for you. We'll give you everything you want. So this is rich. And the idea here is we want to put you in a very wholesome nutritional environment so that as a cell, you can kind of lick your wounds, heal yourself up, because we want you to be strong and viable. But guess what? You're still holding all those mutations we just induced in you, right? So that's what we want to do. So that's basically your recovery. And then what you do is you screen for your mutation. Now remember, we're interested in arginine mutations. So we had two different levels of screen. Number one is we plated our cells that we mutated on media, but this is minimal media, right? So if you are essential, if you had essential gene mutations, that is to say you need these guys in order to live, you don't live, right? We know that amino acids are essential, so we know that the one we're interested in should not live on minimal media. However, that's not necessarily arginine, is it? You got a lot of essential genes. It just says you're necessary for life. But how do you know that this is actually arginine? How do you know it's not lysine or aspartate or some other amino acid that you're looking for? Well, the reason why you know is because the next piece of your selection screen that you do is you follow it up by doing what's called a rescue. So you take your minimal media, which we already know does not support life, and then you add back arginine. And then if you get growth, what that means is you got an arginine mutation. So what does that mean? If the reason why you died is because you don't have arginine, then if we give you arginine, guess what? You're going to live, right? So that's a rescue experiment. What that does is it tells you what the problem was, right? What you were missing. And what we're wanting is those cells that are missing arginine, because that arginine is going to be missing for any number of reasons. So if there's a whole bunch of genes involved in the process, then any one of those genes, if they get fouled up, could cause this problem, right? So now here's what they did. And you got to follow me on this one. You got to follow closely. Here's what they knew and what they didn't know. Number one is they knew that these chemicals were involved in arginine. That is to say, they knew that ornithine was involved, citrulline was involved, arginosuccinate was involved, and of course arginine. And they knew that the beginning of it all was glutamate. But what they didn't know is what the order of it was or how they worked. Okay, so they didn't know any of that. And so what they wanted to do is they wanted to construct a multi-step pathway. Then which is step one? Which one is step two? Which one is step three? So that's what they're trying to do, put these things in order. And they know they're involved. Here's the idea. So they did basically what is kind of like what is a sequential or a serial rescue experiment. So they've already done arginine, right? So that one they've already done. That's part of their screen. So they only pick the ones where arginine rescues them. What does that mean? That means arginine is the thing that's missing and the reason why they're dying. So in all of their samples, they notice that they've got growth, 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 growth. Good. What that tells you then is that if I give you back arginine, then you live. That means that arginine was a thing that you're missing. Now, here's the thing. What about the others? Okay, well, let's go ahead and say, well, wait a minute now. What if I give you arginosuccinate? If you're able to live, if I give you arginosuccinate, then that means whatever is here in the middle, converting arginosuccinate to arginine, you have. By the way, if you don't live, that means that you're deficient in that step. So what did they see? They saw growth, 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 and then here they saw no growth. What does that mean? That means in that particular group, you have a mutation in that enzyme right there. So there's a fundamental foul up in that reaction, that terminal step. Now they kept doing the same thing. They did the same thing with citrulline. So they say, well, if we add back citrulline, does that rescue you? Well, it only rescues you if this enzyme and this enzyme are working, both of them, right? So that was true for this one and this one, but in this one, no, right? So that basically meant there was a fundamental breakdown in the enzyme that converts citrulline to arginosuccinate. Now, in this particular individual, if I give you arginosuccinate, can you finish out? The answer is yes, right? Because there's no problem with this one here. It's this one that's fouled up in this particular situation. Then they did the same thing with ornithine. They said, okay, if you give you ornithine, can you finish out? And the answer was yes in one, but no for the rest. What does that mean? That means there's a fundamental mutation in that enzyme right there. So it doesn't matter how much ornithine I give you, you can't go forward to get to arginine. But if I give you citrulline, guess what? Will you survive? Sure you will, because you were growth there, right? If I give you arginosuccinate, will you survive? Sure, because I gave it to you and you survived, right? So that's basically how they decided what the order was. So you can see, and then of course they did the same thing with glutamate and they realized there was another enzyme up there, right? And so here's basically what they were able to construct. They were able to construct essentially this biochemical pathway where you had 4 different genes. So if you had a mutation in gene H, which encodes enzyme H, then you had a fundamental breakdown in that last step. If you mutated gene G, which killed enzyme G, then you had a breakdown in that step. If you mutated F, which killed enzyme F, then you had a breakdown in that step. And then finally, if you mutated E, you would kill enzyme E and you'd have a breakdown in that step. How do you know that? Because if you had a breakdown in this step right here and you added ornithine, that would rescue. Nobody else is having a problem because you're able to finish out to arginine if I give you ornithine. The problem is that first step. whatever it is that's converting glutamate to ornithine, you don't have it. The important thing about Beadle and Tatum is that they basically taught us how to study biochemical pathways. Of course, along with the one gene, one enzyme thing, right? Because that's what they had. They mutated genes, but they saw dysfunction in enzymes. So one gene, one enzyme. This is also important because with biochemical pathways, as it turns out, when we first did this experiment, what we thought was that you had kind of like one gene, one process, right? So if you had some sort of a process, whether it's replication or transcription or translation or whatever, we thought that there was one gene involved in that. So you had one gene per process. And then on occasion, with a really, really scary process or a really involved process, you might have multiple steps. So the idea was that most processes were single protein, and that on occasion you would have multi-step processes like a biochemical pathway that would require you to do this. However, as we learned more and more about these proteins, what we realized is that's not the case. The biochemical pathways is actually more the rule than the exception. So Beadle and Tatum's work was really instrumental in teaching us how to uncover and discover biochemical pathways. Okay, so let's move on then to central dogma. Now we're talking about probably the late 50s, early 60s. So this is basically something that Crick came up with. And to his credit, even though he still has the devil's beard, and he will still have the devil's beard. I mean, to his credit, he did actually make some important contributions to this particular era, transcription, translation. So credit what credit's due. It's just not due to him for the structure of DNA, right? But it is for this. See, I'm fair. I'm not out of control here. So basically, Crick came up with this idea of central dogma, which is essentially following the flow of information, which we notice is flowing from DNA to RNA to protein. So that's the flow of information. So when you're basically taking information from DNA and you're making it an RNA copy, that's a process called transcription, which I abbreviate XPN. When you go from RNA to protein, that's translation, which I abbreviate XLN. So if you see those abbreviations, that's what I mean by that. And that's generally how things work, right? So you basically go from DNA to RNA, from RNA to protein, and that's the story of this chapter, transcription, translation. There's only one violation to this that we know of, that's retroviruses. Why? Because retroviruses in a genome have an RNA genome. And so what they do is they basically go from RNA to DNA. It's a process called reverse transcription. Okay, but that's typically isolated to retroviruses. But for the most part, the rest of the world moves through central dogma. Now, this for you picture people, this is your picture. So you got transcription going from DNA to RNA, translation go from RNA to protein. So that's just like a picture you got. Let's take a look at the two of these, right? So transcription is important because obviously this is going to be the synthesis of RNA, but generally speaking, you're only using one strand of DNA is read for transcription, not both. The other big difference is obviously when you're making RNA, you're utilizing uracil instead of thymine, right? Because uracil is in RNA, thymine is in DNA. And then, of course, you use this mRNA to direct the synthesis of your polypeptides, which is the process that we're going to see later on in the chapter that is translation. Okay, and this is all happening in the ribosome. So what we'll see is the actual mechanism of transcription and translation, the activity of what's happening inside the ribosome itself. Okay. Now let's take a closer look at RNA, since RNA is kind of at the heart of all of this. Now, originally, we used to think that you had three major types of RNA. That is messenger RNA, which is essentially carrying the code for making a protein. That's why it's called messenger, because it is the message. It's carrying the protein message. The ribosomal RNA, we've already talked about a little bit, right? This is the structural component of the ribosome. We'll see that. And then, of course, tRNA, basically its job is to translate the code in messenger to amino acid. So oftentimes I'll refer to transfer RNA as the Rosetta Stone, which is what? Anybody know what the Rosetta Stone is? That's the program, the original Rosetta Stone. Or was that what you were talking about? Yeah, right. So the program that we think of, like learn Spanish in, you know, half a day or something like that, right, with Rosetta Stone, that's actually named after the actual stone itself, which is an article that was critical in the 19th century because for the first time it allowed us to be able to decipher hieroglyphics, right? Because we had no picture-based language in existence, so we had no idea what the eyeball meant, right, or the cow or whatever, or the standing man. Or we didn't know what these things meant. We knew they meant something, but we didn't know what they meant. But the Rosetta Stone was a stone that was probably made late in the probably last century, first century BC, more than likely, where there's an overlap of Hellenistic culture, Roman culture, and Egyptian culture, which would put you right around that first century BC, when largely the world was still Hellenistic. Thank you, Alexander the Great. Where the Ptolemies, who were Greek, were in charge of Egypt, and the Romans, who were the reigning empire at that time, were basically in charge of North Africa, thanks to their Punic Wars, or the winning of their Punic Wars. So basically what they did was an inscription of some sort of inscription that had three languages on it: Latin, Greek, which was essentially the universal language of the day, because of Alexander the Great and Hellenism, and then hieroglyphics, because it was referring to an Egyptian thing, and it was basically kind of you know, a nod to Egyptian culture because they were Greeks in charge of Egypt. They were foreigners. And so because we knew Greek and Latin, we were able to decipher the hieroglyphics. So we used the stone to translate from one language to another language, just like we use tRNA to translate from nucleic acid language into amino acid language. Okay. Since then, in the last 20 years or so, we've actually discovered lots of different types of RNA. So small nuclear RNA is basically, you'll see this in splicing. We'll talk about that mechanism in eukaryotic RNA modification. There's also small recognition particle RNA, SRP RNA. This is basically for protein localization. So this is how you know how to get a protein from one place to another. And the microRNAs, which there's a lot of them that we're noticing lately, are largely regulatory. So there's like a ton of discovery that's happening in the last 20 or so years. The world of RNA is really blooming, and our knowledge of its function is really blooming. Things we used to think were just degraded scraps of mRNA or tRNA are actually microRNA that have a regulatory function. So there's a lot of good stuff going on. Of course, then, since you have the code in messenger RNA, we're always really interested in code breaking, right? So the idea was, what about this genetic code? Let's break it, right? So involved in this journey initially was Crick. Of course, this was an important thing because a lot of people felt like, okay, if we figure out the genetic code, this may be another Nobel Prize coming, and it may be. I don't think anybody actually got the Nobel Prize for it. But Sidney Brenner, my boy, he's the guy who developed C. elegans as a model organism. So Crick and Brenner did a lot of work when they were trying to look for how DNA encodes the amino acids. One of the early discoveries they noticed was that the code is actually written in 3-letter words called codons. Now, how did they figure that out? They figured that out by working with what's called frame shift mutations. So frame shift mutations are insertions or deletions of either 1 or 2 base pairs, something non-3, right? And so what they notice is that if you basically add 1 or 2 or delete 1 or 2, what you get is a shift in the frame. So let's go ahead and just write some sequence. There, just like that. They were saying is that normally in frame, you would read this in 3s. So this would be 1, 2, 3. So that'd be your first word. 4, 5, 6. That'd be your next one. 7, 8, 9. That'd be the next one. 10, 11, 12. And then 13, 14, 15. So you'd read it in 3s. Okay? And that'd be a frame. So whatever A is would be a particular amino acid. So then how did they figure this out? So let's add one first of all, right? So what if I added an A over here at the beginning? What happens with that? So what they noticed here was that the frame shifted. So now all of a sudden, this first A that we added is 1. This is 2. This is 3. This is 4. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and so forth. So notice in this particular case, your first amino acid is the same, but not your second, right? So what they noticed is that the frame is shifted in that direction. This is basically if you insert. But what if you delete? Well, let's go ahead and delete these two here. And then what do we get? Well, we get a shifting of the frame again. Only the frame shifts in this direction. So now it's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So notice now your first is AAC, not AAA. This is what happens when you have a deletion. Well, they notice that you're reading it in threes. Why? Because you sort of screw everything up whenever you shift it, either 1 or 2. And so this is kind of what they saw. So here is basically your sequence. We're going to take a look at ACG right there. We're going to delete that C out of there. And what we get then is AGC is going to be the new read. But notice what happens. Methionine, proline, and then threonine, histidine, arginine, asparagine. But then what happens is you get methionine, proline, and then all of a sudden it just turns into what? Just a bunch of random nonsense, right? Serine, threonine, alanine, threonine, histidine. It's like, where is that coming from, right? So that's because you shifted it. And now it's becoming nonsensical. So this would be an example of a frame shift. True if we have 1 or 2 base pair additions. But what about 3? What if we do 3? Well, 3, you still get a mutation, but it's not a frame shift. So in this case, notice you're going to take out the entire ACG. So what you do is you go you have methionine, proline, but now this says ACG is missing. You go straight to the next one, and that's histidine. So notice this is all normal. The only thing is you're just omitting threonine. So with that would only be true if you're reading these in threes, and you just omitted it when you did a 3 base pair deletion. You omitted it. So that would be less severe. That's good. The other thing is, basically, we wanted to know is the spaces. Like when you delete something, do the spaces count? That's what we wanted to know, right? So for instance, like if I have something like this where I deleted that one side, does this still count as 1, 2, 3? Or now is it shifted and this is 1, 2, 3, right? That basically means does the space count? And that's kind of what they did here. So here's an example. This is just using 3-letter word sentence. In this case, this is a situation where the spaces count. So this is what we'd expect to see. So if we delete out the T, then we'd say the space still counts. So this would still be 7, right? And so you'd still read it that way. Or in this case, you have the spaces don't count. In this case, when you delete that one, then the H becomes 6, 7, right? Now it's shifted. And you kind of get all those nonsensical words, right? So what we noticed is that it's this pattern right here, right? That the spaces don't count. Okay, so now that we have codons, these 3-letter words, obviously the major question was, okay, great, but how do we, let's go start assigning these words to meaning, right? So which words assign for which amino acids, right? That's the next big one. And Marshall Nuremberg did a lot of that. So basically what Nuremberg did is basically created an in vitro translation assay. What that basically means is probably just a bunch of cell guts, cell slurry, cell extracts, right? And inside this test tube is everything you need in order to do translation. All you need is the template, the mRNA. And Nuremberg supplied that, basically created it and then supplied it. So here's what Nuremberg did. So basically made his own template, let's say just a bunch of U's. And then he fed it to his little translation mixture. And then what did he get on the outside? 3 phenylalanines connected together. So now, as a confirmation of the frame shift data that we just saw, how many phenylalanines do we have? Not a trick question. Yeah, it's 3, right? How many nucleotides did we feed this? Yeah, 9, right? So 9 divided by 3 is what? So that means that you have a 3-letter word. So basically, Nuremberg's work confirmed the 3-letter word codon idea. So what this also does is it basically says, guess what? U equals, got a very simple, straightforward experiment. All you have to do now is just make a bunch of sequences, right? Well, let's try UUC and CAC and AAA and AUU and I mean all sorts of things, right? So eventually you're going to get this big pile of assignments. And he did that. He also got a couple of interesting things, right? For instance, he also found stop codons. Whenever he found UUA, UGA, Go Bulldogs, or UAG, he noticed those are stop codons. So for instance, if he fed his system this one, what did he see? He saw not 3 phenylalanines, but 2. Why? Because that one is a stop codon. It tells the translation machinery to stop. And he found 3 of them. He also found a start codon. So this was basically common to all. And it encodes a methionine. So this is kind of like where you start your translation. It's like your start sign. Thing you also notice is the code is what's called degenerate. And that's a big fancy term for essentially to say that some amino acids are actually encoded by more than one codon. And then when you take all of his data together and put it all together in a big table, then this is what you get. This is your code. Representing mRNA, basically what you see is the first letter, which is read straight down the column. The second letter is read across, and the third letter is read down this column. So for instance, if I have AAC, that is going to be A is in this column. A is going to be in this one right here. So it's this block right there. And then C is going to be right here, which is asparagine. So then one of the good things to do with this particular table is challenge yourself. So I'm going to write some sequence here. It's going to be DNA. The first thing you want to do is replication. Make the complement. How do you do that? What's the complement to 5'? It's 3', right? And the complement to A? Remember, we're doing replication now, right? So you fill it in. T, 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 C, G, G, A, 5'. Now what you want to do is take this strand and do transcription. Transcribe it, right? Again, now this is RNA, right? So what are you going to do? 3', U, U, A, right? G, G, U, 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 C, G, G, A, and 3'. Excuse me, 5'. This one up here at the top. There. Now, the important thing, the reason why I did that is because remember the polarity of DNA, right? That anti-parallel thing going on. Also remember that whenever you see sequence written down, it's always by default written 5' to 3' going left to right. So when you look at your genetic code, when I see CUU, this is the 5', this is the 3', always. Notice how my mRNA now is flipped around. So in order to translate this, I would need to rewrite this as 5' AGG, right? GUU, UGG, AUU, 3'. And now what you want to do is translate. So what's AGG? So AGG would be arginine, right? Translate GUU. That's there, GUU, that would be a valine. How about UGG? That's a tryptophan. How about AUU? It's an isoleucine. So what you've done is replication, transcription, and integration by doing that, using your genetic code. If I were you, I would practice that. Just make up some random DNA sequence, replicate it, and then transcribe it and translate it and get good at doing that procedure. That's easy. Wait, which one are you talking about? Where? Right here? That's because that's this upper template. That's the transcription using that as your template, the top one. So A will give you U, A, U, right? Because it's RNA now. You're doing transcription, not replication. So basically that would be UUA. But when you write it reverse, remember, because it's backwards, you have to flip it around. So you have to rewrite it going this direction. That's what this guy is down here. Okay. So practice that and kind of get used to that idea. Okay. Yeah. After I did this?
Yes, when you did 3 to 5 again, you don't go back and then do 3 to 5 again.
No. No, because that's your mRNA.
Yeah.
That's your code. So from there, remember the flow is DNA to RNA. We just did that. That's transcription. And now you've got the RNA or the mRNA. And so you're turning that into protein. And that's essentially what's happening when you're translating it using the table. That makes sense?
Are we gonna have to remember all of the ones in the bottom?
No, you don't have to memorize the table. So like something like this, like if there's a question on there, like, you know, here's a sequence, transcribe this and then translate it, you would have this table, right? It doesn't make any sense to try to memorize this. That's just a real waste of brain space. Okay, let's go to cute little pig, shall we? So this is kind of like the idea where the code itself is universal. That is to say that all organisms share the same ACGT code breakdown. And there's actually more similarity than there is difference, which is kind of wild, actually, if you think about it. But hey, when you got a good system, why reinvent the wheel, right? Just duplicate success. That's what we're talking about here. Now, this has been helpful for us, largely because it's allowed us to do genetic engineering, which I know sounds very scary and controversial. But what we're talking about with genetic engineering is actually something that's saved us quite a bit. So our ability to actually understand how genes actually work has come as a benefit of this. If we haven't been able, if the genetic code wasn't universal, then we wouldn't be able to use mice in order to understand how to develop a therapeutic in humans. We would have to use humans. And that would look very scary if we had to use humans to experiment on in order to generate therapeutics, right? Because, you know, who gets to be that one, right? Who gets to be the guinea pig on that one? Usually that doesn't go well when we try to do that sort of thing. But it's allowed us to basically do and study genetics in other systems so that we can learn about how genetics works. And this is kind of like the big thing that Mendel didn't realize, but what really turned out to be a big thing, right? The reason why Mendel is like the father of genetics and why what he did was so important isn't because he helped us understand what was happening in pea plants. It's because what was happening in pea plants was a universal truth for what happens in all organisms, because our code is all universal. We all basically have this same system in place. That basically means I can learn something in one system and apply it to another. That's basically what we use genetic engineering for. So here's a good example, right? So it allows us to be able to do what's called transgenics. And transgenic, so trans across genix, the genus, so that's different organisms. So this is a transgenic pig right here, and this is a normal pig. And generally, transgenic, we do kind of a cute little molecular trick. I'm going to explain how genes are basically organized. I did this actually a little bit already, right? Because we talked about tumor suppressors and oncogenes. Remember that one? So basically, you're going to have this idea where you have this protein, and it's driven by some sort of a promoter with some regulatory region just upstream of it. Remember that one? So in this particular case, what we can do genetically is, it looks like in this case, what they did is they picked a gene that was probably like a skin gene, maybe like a keratin thing. So it's basically wherever there's keratin, it glows, right? That's kind of a ubiquitously expressed gene in the skin. So what they did here was, instead of the skin gene, is they remove it. And in this pig, they do something different. They replace it with what's called a reporter. And a reporter gene, in this case, is something we can see, right? So this is probably something like a green fluorescent protein, which is the protein that makes jellyfish glow. When we shine a black light on it, it actually fluoresces green. So wherever that protein is being made, it'll glow. Now, how do we know where it's going to be made? Remember, the promoter determines where and when the protein is made. Notice it doesn't actually know what it's making. It doesn't do a check down and say, oh, wait, you're a skin gene, or no, you're not a skin gene, you're a GFP. It doesn't do that. It's whatever you connect to a promoter, it will make. It just turns things on, right? It doesn't determine what the nature of that is. So when we put this reporter like GFP behind this promoter, then instead of making skin protein, it'll make GFP. Every skin protein would have been made. As a result, when we shine a black light on this little pig, then it'll glow wherever that original skin protein would have been. And that's the reason why you see yellow hooves and yellow snout and kind of yellowish fur. That's been a really, that's allowed us then to be able to really study the distribution and the role of proteins, because we get a lot of information from what's called localization studies. That is, say, I've got this protein X, but I want to know where it is. Thank you. We're back. Okay, everybody can still hear me okay? Okay, so like I said, one of my favorite experiments was using luciferase, which is an enzyme that makes fireflies glow. So there's a substrate that'll break down and it'll create light. And so that's kind of what makes the little fireflies' butts glow. What they did was they took this luciferase and they actually put it into a ubiquitously expressed promoter in tobacco plants. And what they ended up getting was a glow-in-the-dark tobacco plant. It was like cool. It's like the entire tobacco plant was like outlined, like glowing. It's actually pretty amazing. But that allowed us to be able to see where the distribution of that protein was. And so we've been able to learn a lot from that. The tobacco plant is fine. It's like, it's happy. It doesn't have that protein, but otherwise it's happy. I mean, it's just doing its thing. You turn off the lights and you get a glowing tobacco plant. It's actually pretty cool. You can actually Google that, glowing tobacco plant. So basically we take a look at transcription. We're going to start off first of all with prokaryotic transcription, then move into eukaryotic transcription. So when you take a look at transcription itself, right? So you're basically making mRNA. And now remember, RNA polymerases don't need help, right? These guys are self-starters. So you don't need a primer to do transcription, but what you do need is a promoter. We're going to talk about that one here in a second. That's PowerPoint, that one. Don't worry about that one, right? You're going to need a promoter. We already talked about that one a little bit, right? You're going to need a start site, which is actually not part of the promoter, but this is basically the first piece where it says, okay, start your transcription here, start your mRNA here. And then of course you need a termination. So you need a place to stop. Notice the three-step mechanism. There's initiation events, there's elongation events, and then there's termination events, right? So the same thing is true here. So let's take a look at the promoter. So when you take a look at the promoter, the important thing is that the promoter itself is the one that recognizes itself. So basically the promoter is responsible for binding the correct polymerase. So that's all found in the promoter. So the promoter is the one that's specific to make sure that the right polymerase gets to that site and does that. We're going to take a look at some RNA polymerases here. They're not all the same, just like we saw with the DNA polymerases. They have slightly different jobs. So the promoter is slightly upstream of the start site. This is a non-transcribed region, which basically means it's a regulatory region. So only stuff that is post-start site is transcribed. So this is your site of initiation. Right? So you take a look at it. Take a look at the core RNA polymerase. We see is a multi-subunit, what's called a holoenzyme. That just means a really big complex. That's essentially what that means. Multiple subunits. The big one that you want to focus on, however, is this guy down here, the sigma subunit. You can see the sigma subunit actually looks like little pinchers, like an index finger and a thumb. So with that, basically what's going to happen is your sigma subunit is literally going to grab onto or pinch the DNA and bind to the DNA. With the index finger area, it's designed to bind to a specific region called the Tata box, which is at the what's called the -10 region. Now, there's a bit of nomenclature here we need to sort of weed ourselves through. So the start site is considered +1. That is to say, the first nucleotide that makes it into the mRNA. As you start to move downstream, the next nucleotide is +2, the next one after that is +3, then +4, then +5, and so forth. Now, what about the opposite direction? What about this side, the non-transcribed side of the start site? Well, this is where it becomes minus, right? So the next nucleotide upstream of the start site is -1, and then the next one is -3 and -4 and 3 and 4 and then 5 and so forth, all the way up. At -10, you find the Tata box. Now, the Tata box is what's called consensus sequence. which means it's not variable. That is to say that it is a TA rich region that is ubiquitous among all taxa. This is sort of like a flag that says, bind your index finger here. This is one of the binding spots for the polymerase, okay? That's the reason why it's consensus. It basically never changes. It's the same from gene to gene and from bacteria to bacteria. Now, further upstream, where the thumb part of the sigma subunit is, it will basically grab onto the -35 area, which has sort of a TA-rich region, but it's not quite as tight in its conservation as the Tata box. So the Tata box is really tight, but this region is a little looser, but it's at the -35. So what's happening then is your polymerase will come in, bind to the DNA at the -10 site with the index finger, and at the -35 site with the thumb, okay? Now, once it has a hold of that, then it's ready to clamp down, and it'll start unwinding the DNA, and it'll start making the actual transcript, setting down the first nucleotide at the start site, which is the +1 site, and then off it goes. So once it actually binds to that and the sigma subunit has actually found its correct location, then what happens is it dissociates, and then the polymerase is off. So this is basically all initiation. Getting your polymerase bound, getting your DNA unwound, then once your sigma dissociates, then you start to segue into elongation events, right? So once you're done, then your RNA polymerase will grow in the same direction as DNA polymerase, 5' to 3' direction, adding instead ribonucleotides instead of DNA nucleotides. However, the important thing is that within the polymerase itself, it's going to have the open template, and then it's going to have your growing RNA transcript in what's called a transcription bubble. The important thing about it is that the bubble doesn't last. So basically, after your transcription passes, it basically closes up the bubble. So when you see this, what you'll notice is it unwinds here on this end. It'll basically lay down the RNA, and then before it leaves, it'll rewind it on the back end. So what you really see, like if you were to look at an RNA polymerase from the outside, you would see double-stranded duplex DNA going into the polymerase. You'd see the same thing coming out, because the replication bubble, the transcription bubble only exists inside the actual RNA polymerase. So it's very simple. You notice the same mechanics are true for replication, right? You have something to unwind it, you have template, and then you just basically pair it up and match your nucleotides to it. When you're done, wind it back up, you're ready to roll. You basically replace the way that you found it. What about termination in prokaryotes? So basically, termination in prokaryotes has actually got a sequence signal that actually stops it. This will then cause the dehydration synthesis to stop. You'll have a little bit of a DNA RNA hybrid, and then what will happen is the RNA polymerase will let go of the DNA, your DNA rewinds, and essentially you form a hairpin. Now, a hairpin is basically just a tight loop that protects the single-stranded RNA. Because remember, the fate of all single-stranded nucleic acids is degradation. So what do? You basically form a hairpin like this that protects your single-stranded DNA. This is what it looks like. So here you can see you're finishing, and then you kind of form this hairpin structure. So you can kind of see if hairpin, these guys are not complementary to each other, so it forms like a little bit of a loop there. But then otherwise, this is all protected. And this also kind of structurally causes the RNA to dissociate. And so here you can see the dissociating polymerase. It's a letting go of the mRNA. The DNA rewinds itself. and then this goes off to do its job in translation. So the important thing about prokaryotes, because they don't have any organelles, they just have, it's all open in there. When they do their transcription, there's no reason why they need to wait for their transcript, their mRNA to finish before they start working on it translation-wise, before they start decoding it. So as a result, in prokaryotes, you'll see the transcription and translation are coupled. They're happening at the same time. So for instance, when you see this, before the transcription is done, the ribosomes will actually grab onto the transcript and start translating. We're gonna see a picture of that here in just a second. So one of the things that this is helpful for is because of the structure of bacteria in particular, the way they sort of structure their genes into structures called operons. So these are basically groups of functionally related genes. So here's a good example. So they're basically regulated together. That means they have a common promoter. So here's what an operon looks like. Let's imagine you have 3 genes in a biochemical pathway, A, B, and C. And so you need all 3 genes to be active in your biochemical pathway. So bacteria will organize this in the form of an operon. So they'll give you one promoter here that drives all of those. So basically what happens is when you fire off this promoter, you're going to make a transcript, an mRNA, that is called polycistronic. That is to say that the information for all 3 genes is on that one mRNA. It's like for the price of 1, you get 3. That's what an operon is. Now, how does it work in eukaryotes? In eukaryotes, it's different. Let's say that we have the same biochemical pathway with 3 genes involved. What we do is we have A under its promoter. We have B under its promoter. And we have C under its promoter. Separate. So what does that mean? That basically means you have more control. So you can nuance your response, right? So you can regulate separately A, B, and C. If you want a little bit more A relative to B and C, you can do that with a eukaryote. You can't do that with a prokaryote. If you want a little bit more B than A and C, then you can do that by upregulating B. So you can have differential control of each of these, okay? And that's kind of common for eukaryotes. They have that kind of separated control and regulation, which gives them the ability to modulate protein expression a lot more finely than what bacteria can do because they're all based on operons. But the idea is that because you have basically all the genes you need on one mRNA, why not just start translating it like as soon as you get something, even before it's done transcribing? And that's exactly what you have here. Here is the image of this coupled transcription translation. So here's your DNA, and this is your polymerase, and then your transcript, your mRNA transcript is coming off your polymerase. But notice what happens. You're not even done transcribing your mRNA yet. And what's happened is you've got ribosomes that are already glomming on to the end of your transcript as it's coming out, and it's already starting the process of translation. So you can see an incomplete transcript, which is still being transcribed, is still ongoing. And now you have translation happening. So all your ribosomes have already glommed on to the finished end of the transcript and is already starting to make proteins. So you're coupling this. So basically the two are happening at the same time. Why? Because it's all happening in the same space. By the way, why isn't it coupled in eukaryotes? Very good reason why. Transcription happens where? Like local, like in the cell, localized wise, right? Where does transcription occur? Where's the DNA? Yeah, right? Because you're making a copy of the DNA. That basically means the transcription is happening in the nucleus. Where's most translation happening? Where are the ribosomes? Are they inside the nucleus? They're outside the nucleus, right? So notice what you have. Transcription, which is happening inside the nucleus, is separated from translation, which is happening outside the nucleus. This is the reason, biggest reason why eukaryotes don't couple transcription and translation. But what they do have to do is they have to make it inside the nucleus, and then they have to transport that out to the ribosomes, right? We'll talk about that. So let's take a look at eukaryotic transcription. Obviously, there's a lot more bells and whistles here. Instead of one polymerase, we have three. So the first polymerase, Pol-1, its job is to make ribosomal RNA. Pol-2, which is the one we study the most, is the one that's making mRNA and some snRNA, or that's the one that's involved in splicing. And then Pol-3 is going to be making tRNA and some other small ones. But how do you know which one is which, right? So for instance, how do you know a stretch of DNA is encoding a ribosomal RNA versus a tRNA versus an mRNA? Well, the differentiation is the promoter. The promoters provide the specificity and recruit the correct polymerase. So how does a Pol-1 and a Pol-2 and a Pol-3 know that it's binding to the promoter of a ribosomal RNA, tRNA, mRNA? It's the promoter that provides that specificity. So the tRNA promoter will recruit Pol-3, not Pol-2. Okay, so that's where that recognition comes from, is from the promoter. This is the reason why promoters are so important. Matter of fact, this is one of the things we learned. The gene expression, that is to say the pattern of how genes express themselves in making proteins, is not driven by the gene itself. It's driven by the promoter in the regulatory region. That's really what does it, okay? So let's take a look at the initiation events of transcription. So number one is in eukaryotes, we have a lot of transcription factors, a lot of them, right? I mean, obviously we have Pol. Now, most of our studies with transcription has been polymerase 2. So most of what you'll see is for Pol-2. But basically Pol-2 will be recruited and will bind to the promoter to initiate your gene expression. But ultimately it's the transcription factors that recruit it. And then of course you have termination events, which we don't really understand very well in eukaryotes. We do know that it terminates, but it's not a sequence thing the way it is in bacteria. So it's a little bit different there. So let's take a look at some of the initiation events. So first of all, it's recognition, right? So recognize the promoter. And we have transcription factors, TFs. The first transcription factor is something called TF2D, which stands for transcription factor 2, that's for Pol-2, and D, there's a whole bunch of them, right? So TF2D actually has two different domains to it. It has a transcription factor binding domain, where other transcription factors bind to it, and it's called what's called a TBP, a TATA binding domain. So the job of TF2D is to do what? To identify the TATA box. So it binds to the TATA box and identifies it, recognizes it with its TATA binding domain. Then with the other parts of the protein, it will recruit and bind to other transcription factors. So you can see all these transcription factors now are coming in, and they're going to be binding to TF2D. And so here you can see the transcription factors are all bound and tethered to TF2D, and you have lots of different ones. And then eventually this will recruit your polymerase to that site. So those transcription factors are recruiting the correct polymerase to that site, and it'll bind to that site with the binding to these transcription factors. And this forms your initiation complex. So all this right here are events of initiation and transcription. Once your polymerase is now in place, you can go into elongation. right? So, and we've already talked about elongation. So once you have this, then you can go into elongation. And like we said, we don't really understand termination. We do know that it does stuff. Okay. So the previous slides, when we saw the transcription bubble and how it's moving along, those were largely the same in eukaryotes. That was largely elongation. Okay. In eukaryotes, this is how initiation works. So there's a lot more going on in there. And as a matter of fact, transcription, there's a lot. This is just like, this is like the sock puppet version of transcription. I mean, there's a lot of things going on in transcription. It's a very dynamic process. At the end of it is, you go through elongation, you make your mRNA, you hit the termination segment of it, and then you fall off, and you have this mRNA now. But what shape is it in? So your mRNA at this particular point is in what's called a primary transcript. So this is what's called pre-mRNA, premature mRNA. So your mRNA is not ready to be transported out of the nucleus. Why? Because number one, it's single-stranded, which means what? You're going to be degraded on both ends, aren't you? Those exonucleases are going to take a look at you. They're going to be like, oh, hey, I'll go ahead and chomp you down from the 5' end and from the 3' end. So one of the first things you have to do is basically processing, mRNA processing. This is only something that happens in eukaryotes. It doesn't happen in prokaryotes. But one of the major things in mRNA processing, once you make your mRNAs, you have to basically modify it now in order to protect it and get it ready to be read. The first thing you do is you add a 5' cap on this end to block and protect it from degradation. It's also involved in translation. This is basically where the ribosome binds the mRNA. By the way, okay, so you've blocked it now at the 5' end. But what about the 3' end? You can still get chewed down from there, right? So you got to basically protect that end too. So you have to create what's called a poly-A tail. So this poly-A tail apparently is just not very pleasant for exonucleases. This is a problem with that one. So this will protect it from degradation, but it's also a signal for other processes. So there's also a little bit of regulation involved in this poly-A tail as well. And then the third major thing you have to do, so that's protection for the most part. And the third big major thing that you have to do is you have to basically prepare what's called an open reading frame. Now, what is an open reading frame? An open reading frame is basically just essentially a continuous region that will be turned into amino acid. Think of it this way. What your mRNA is like now is like a sentence, like a 3-letter sentence, right? Like we saw before, the fat cat ate the fat rat, right? But now what's happened is instead of nice clean spaces, what you have now is like you write the first word, the, and then what you do is you pound the keyboard and you create a bunch of random letters, and then you write the next word, fat. And then you pound the keyboard again, and then you write the next word, and then you pound the keyboard and you write the next word. So basically what you have is you have your words, intervening in between these are a bunch of sequences that aren't part of the sentence. That's kind of what it is right now. So these non-coding parts of the mRNA, what's called introns. So this is the kind of the random, it's not actually random as it turns out. These are the random portion or random sequence in between coded regions. As it turns out, it's not actually random, but it looks random, right? So it's non-coding. The exons, these actually are the coded regions. They will actually become amino acid. you have really is if I draw like a box here, so here's exon 1, there's an intron, there's exon 2, intron, exon 3, an intron, exon 4, an intron, and there's exon 5. So what's the open reading frame? The open reading frame is basically you taking that sentence and deleting out the random letters in between the words. So what do you have to do to clean up that sentence to create an open reading frame? Delete the key poundings in between the words, right? And put the words together so that you can read the sentence in an open reading frame. You have a start spot, don't you? The capital letter. You have an ending spot, don't you? The terminal punctuation. Between the capital letter and the terminal punctuation is your open reading frame. You can start from the capital letter and you can read without interruption the sentence. The fat rat ate the fat cat. Well, that's a big rat. But anyway, that's essentially what you've got coming off of that. So you have to modify that, right? You have to create that open reading frame, and we do so by a process called RNA splicing, which is done by a protein complex called the spliceosome. So here's kind of what it looks like. So here's your mRNA. You create your, then you've got your, in this case, it's a methylguanine phosphate cap that you use. So that's your 5' cap. And then in between, you've got a series of exons and introns that you have to process now. Okay. So let's take a look at how you do this process. So here's an example, right? So here's your mRNA. I'm going to skip from the template to the RNA. So here you can see you've organized it into exon 1, intron 1, exon 2, intron 2, and so forth. But that gives you basically an exon here. This is an intron, an exon, an intron. So how do you cut this out? So all you have to do basically is take the introns and essentially clip them out, and then you have to sew the exons together. That creates one continuous reading frame. So this is an ORF, an open reading frame. You start reading here, and you don't stop without interruption until you get to the stopping end point, right? The terminal punctuation. So what's the process to that? Well, the process is splicing. So here you can see that what happens is you've got basically the spliceosome, which is as a series of what's called SNRPs. Which is a snRNA protein complex. And you can see the snRNAs are basically a template inside these SNRPs. So you have several different ones. You have like U1 to U6. That's how we label them. So U1, its job is to basically recognize this exon intron boundary. Just above or just above the downstream exon intron boundary is what's called a branch point. So let's take a look at a little bit of architecture here of an actual intron itself. And I want to go back to this one to kind of show it because I can show everything. So here's the big question. So if you've got a bunch of exons and introns and it's all just how do you know what's exon and how do you know what's intron? What tells you where the exon is and where the intron is? So they have sequence that tells you that, consensus sequence, right? And they have what's called a 5' splice site. That has consensus sequence. What is that consensus? It starts with GT, and then the other 3 are variable. But there is a consensus GT. So every 5' splice site is marked by a GT. On the 3' side, there's a 3' splice site. again with another consensus sequence, terminal AG. So every 3' splice site has an AG. Now the other 3 that are upstream are variable. But when you see a GT with a downstream AG, that defines the boundaries of an intron. Now, in addition, right about here, just near the 3' splice site is an adenine that's invariable that is referred to as the branch site. We're going to look at the branch site here in just a second. But here's what happens. The spliceosome will recognize the 5' splice site and then the downstream 3' splice site, and it'll cut out that intron. So it'll just cut those introns out, GT to AG, GT to AG, and then it'll sew these exons together. So you get exon 1, exon 2, exon 3, exon 4, and a little nib of exon 5. That's an open reading frame. Now, a lot of times students ask the question, but wait a minute, if it's just a GT-AG pairing, then is it possible for this upstream GT right here to pair up with, say, this AG down there? And the answer is that's possible. So it is possible. So if you pair up this GT with this AG, then what's going to happen is you're going to cut everything out in the middle. And you're going to be left with exon 1, but it's going to skip all the way down to exon 4 and a little snib of exon 5. By the way, you can pair up other GTs and AGs. So this allows you to be able to create alternative open reading frames called isoforms. These are different proteins that came from the same gene. So the good thing about this, by the way, is that for the price of one transcription, you've doubled your productivity. You don't only just make one protein, but from this one, you can make 2 different proteins. That's not bad. That's not bad. By the way, that also reduces the burden on how much genetic material you need to have in order to generate the mind-numbing number of proteins that we have in our body. So what's happening during splicing? Well, the U1, which has an snRNA1, has a piece of its RNA that's complementary to that 5' splice site. So it recognizes the 5' splice site. Same thing's also true for the other SNRPs that are down on the other end. One of these guys, U2, will actually recognize the branch points. And then these 2 will then associate with each other. They'll kind of pull each other together and they'll kind of bind together. That's looping your intron out. And then once this happens, then you basically cut at the 5' splice site. I'm not going to go into the details of this. It actually does get a little messy, but you're going to cut this boundary right here, and you're going to attach this loose end to the branch site, creating a lariat, which then goes off and gets degraded. And of course, at this point, you've also cut that 3' side as well. Now you've got your intron tied up in a lariat, which goes off and gets degraded. Now all you have to do is sew together your exons. And that's exactly what happens. You sew your exons together, your lariat gets excised, your spliceosome falls apart, and then it looks for another mRNA to process. So what about these introns? These are curious little things, right? Before, we used to think of introns as basically junk DNA. That's what we used to call it, junk DNA. Mostly because we didn't really understand it. But when we actually take a look at introns, it's actually quite variable. There's not a lot of consistency to introns and how they work. So for instance, some genes don't have any introns at all. Other genes have buckets of them, 50 or more. I mean, that's a ton of intron. So it's highly variable from gene to gene. We used to think that introns, and we still kind of do think this a little bit, that introns were ancient viral invasions. right? Because a virus will basically have its genome, it'll infect you. And then one of the things that a virus does is it integrates its genome into yours. And so you kind of have a viral genome inside your DNA. Right? And the idea is if it just sort of stays dormant and it sort of loses its oomph, then that becomes intron. Right? That's kind of where we used to think, but it's not, we don't really think that so much anymore, but good.
Is it always
Yep, that's how we define it. So if you see a GTAG with the branch point, those are consensus sequences, landmarks that you see in an intron. So everything else is up for grabs in between there, but those three are solid. So what we actually believe now, right, instead of, I mean, there was a problem with this one, right? Because if you think about it, if you have like 50 introns integrating into your genome, what you're essentially saying is that evolutionarily, you're essentially blowing up a genome. That is catastrophic fail. And I think we've sort of backed off of this one a little bit because the amount of destruction that you would create from this amount of viral insertion would be genomically catastrophic, right? It would be kind of like saying we built LA by dropping a bunch of nuclear bombs. And then it just randomly all came together, right? That there's no way that a bunch of nuclear bombs can have any kind of a positive effect on building a city, right? It's only destructive. And that's kind of what we came to. It's like, maybe if you had a viral integration, we could utilize that to our benefit. But if you had a ton of these coming in, like is what you saw with like 50 of these things in a gene, then what you're talking about is a just complete blow up of the genome. There's no way to see that as anything other than disaster. So now what we're thinking of is that rather than just sort of like a viral invasion, we're actually thinking that these are a little bit more purposeful, you know, more of a purposeful development. So the exons themselves seem to be organized in terms of functional domains. That wouldn't be true if this was just random integration of a virus, right? So the fact that you actually have these introns coordinating or separating off or sectioning off these exons into discrete functional domains seems to suggest that there's a bit more intention in there and that there's more organizational in there. Because a virus integration would be random, and the likelihood of getting that kind of level of organization from a random event across the entire genome is actually preposterously, stupidly zero, right? So this is the reason why we're thinking it's a little bit more intentional in that nature. We can also shuffle exons and kind of get different effects. And this kind of creates a lot of variation, right? And a lot of nuances to that. The other big thing that this leads to is also something called alternative splicing, which a lot of people thought wasn't a thing back in the day. But it's actually a big thing. So what is alternative splicing? It's what we saw. Alternative splicing is basically alternative cut patterns. So when we saw two different isoforms being formed from our mRNA template, that's alternative splicing. That's what that is. So you're creating two different alternates with one splicing event. Alternative splicing patterns. I can get back there sometime before Christmas. All right, so this would be like alternative splicing. Yeah. It creates isoforms by using alternative splicing patterns. Okay. So why is that a thing? Because we used to think alternative splicing was just kind of like not a very big deal. It was sleepy backwater. But what we now know is that actually alternative splicing is a pretty big deal. And actually, it's funny because I was in the meiosis lab at UC Davis. Just next door to us was the Burtis lab, which is still an active lab at UC Davis. And at this particular time, in the 90s, they were working on alternative splicing in Drosophila. And one of the funny things they saw was a protein that they isolated called double sex. DSX. The problem is double sex had two different versions. It had a male version and it had a female version. The male double sex protein was found in males and was the protein whose job it was to masculinize the fly tissue to a male. So all those secondary sex characteristics of a fruit fly, which don't sound cool, they got sex combs on their legs and pigmentation, that was all driven by double sex M, DSXM, the male version of it. In females, the feminization of her tissues was driven by double sex F. As it turns out, when they tried to map these proteins to their individual gene that created them, they all came from a single gene, double sex. So what happened was females had a different version of double sex than males. The reason why, this was caused by alternative splicing. So when the double sex gene was produced in males, their splicing pattern produced DSXM, and they become male. In females, their splicing pattern would produce DSXF, and they become female. This was the first time when we actually highlighted very clearly the powerful importance of alternative splicing, because here it's basically sex characterization in fruit flies is driven by alternative splicing. So it was a big deal. As we started to work with humans and look at alternative splicing, what we noticed was alternative splicing was actually a lot more common than we thought. So for instance, 15% of most of our genetic disorders are splicing defects. They're altered splicing problems. Between a third and almost up to 2/3 of our genes have some sort of alternative splicing associated with it. Think about that. That's a ton of alternative splicing genes. That tells you that the majority of your genes are actually doing alternative splicing, which makes a lot of sense, right? Because one of the things we were thinking of, like when we went into the Human Genome Project in sort of like the turn of the millennium, we were trying to think of like, okay, wait a minute, in terms of the human genome, like how many genes do we have driving our phenotype? We were thinking, oh my gosh, it's got to be millions, right? I mean, think about how many proteins are active in your body. I mean, it's got to be millions of genes, potentially billions of genes firing, because every protein needs a gene to encode it, right? One gene, one protein. So we were thinking it's got to be millions of genes. How many did we actually find? Between 20 and 30,000 genes, actually. So to make your phenotype where you are right now, you're looking at the activity of maybe about 20 to 30,000 genes. Okay, first of all, if you're a genomics person and you're a geneticist, you're basically kind of trying to clean your jaw up off the floor, because this was an overestimation, like by a lot. So this blew us away. How come there's so few genes active in the human genome? 20 to 30,000. That's a lot less than we were thinking. The reason is because of alternative splicing, because you have fewer genes active, but they're all making multiple isoforms. So it's not one gene, one enzyme, it's one gene, multiple enzymes is what it is. And so what you found out was that basically these 20 to 30,000 genes are capable of making tons and tons more mRNAs than we ever thought. Okay. So that kind of highlights it. So that's basically processing. So once we actually do our RNA splicing, we've got our 5' cap, we've got our 3' poly A tail. We've basically created our open reading frame in the middle. And now what are we ready for? Now we're ready to actually put it to work. Let's go ahead and transport it out of the nucleus and let's get it to work inside a ribosome. So that gets us to translation. So that's all transcription and RNA modification. Now we're going to take a look at translation. So we take a look at translation. The first thing we need to do is we need to talk a little bit about tRNA. So we're talking about tRNA, and we're at ribosome in particular. We have to talk about how you actually manage to build the molecule itself. So we already know how to make the RNA. That's transcription, right? We already know the Pol-3 is the one that does that job. Got that one. Okay, there. However, when you take a look at a tRNA molecule, you have to understand that it has two basic member domains. One of those domains is responsible for carrying the amino acid that it's assigned to carry. right? Now, that amino acid is attacked by a very important enzyme called the aminoacyl tRNA synthetase. We're going to take a look at the mechanism of that here in just a little bit. The other part of it is basically the anticodon loop. This is basically complementary to the mRNA codon. So one end of the tRNA has an anticodon loop that's complementary to the mRNA. The other part of the tRNA has an attachment site for the amino acid. Okay, it has to have those two things. So as a result, these are your basic views of tRNA. So you can see it's an RNA, right? But it is highly folded up into all these stems and loops. And these stems and loops, by the way, are important. It kind of makes it look like a little bit of a clover leaf, but they're important because these are the sections that they use to bind into the ribosome, right? So these stems and loops is kind of how they grab onto the ribosome when they're binding into it. But notice you got lots of stems and loops. On one end, you've got your acceptor end, right, which is binding to your amino acid. And on the other end, one of the loops is your anticodon region, which is finding and pairing up with the codon of the mRNA. So here's your ribbon view, your space fill. This is the one this is the cartoon. And we'll basically work with the cartoon through most of the chapter, right? So here's your acceptor end, and then here's your anticodon end down here. So those are your two ends of your tRNA, which is this clover leaf looking structure. Now, how do we build this, right? We already have the RNA piece of it. That Paul 3 did that for us. But now how do we go about attaching the amino acid? That's called charging. So when you charge up the tRNA, basically what's going to happen is it's going to add an amino acid to the tRNA. However, it's going to pair it up the correct amino acid to the correct anticodon. So each aminoacyl tRNA synthetase will recognize one amino acid. It'll be able to bind to multiple tRNAs, but it'll focus on one amino acid and attach it to the correct tRNA. So its job is to add the amino acid. It is going to require ATP, so it does require energy. It's an energetic process. And then it'll basically add that amino acid to the tRNA. Now, the important thing to understand is where the quality control comes from. The quality control, like how do you know that you have the right amino acid paired up with the right anticodon? That's the question here. And who's responsible for making sure that is a correct pairing? The important thing to always understand is this phrase right here. The ribosomes do nothing for quality control. They don't double check on you. They don't make sure it's like, oh, I think this is the wrong amino acid. No, they don't do any of that. All they do is they take what you give it and they add it to the growing chain. That's all they do. So where's your quality control? Your quality control is purely governed and driven by this enzyme here, the aminoacyl tRNA synthetase. This is responsible for quality control. Which is the reason why we talk about this step in a little bit of detail, because this enzyme, the aminoacyl tRNA synthetase, is the only one that is responsible for making sure that your amino acid is paired up correctly with your anticodon. Nobody else does it after that. So if this guy screws it up, then it's screwed up. Okay. So this is a very, very important enzyme. because he's making sure that he's building. Take a look at the activities of this particular. So basically, the synthetase is going to have two active sites. It's going to have an active site for the amino acid that it works with. And then it's also going to have an active site for the tRNA that goes along with it. So the first thing that happens is you get hydrolysis here of ATP, which then allows, in this case, tryptophan to bind to this active site by way of this hydrolysis. So notice you have two phosphates that leave, which means you have AMP left over. That allows you to bind to tryptophan to your synthetase. Then you have the tryptophan anticodon, which then comes in and will bind to its little tRNA site. Now, there's one thing that I want to mention, right? So first of all, obviously, number one, ATP is involved. So you have to have ATP involved here. And then that allows tryptophan to bind. So hydrolysis of ATP allows tryp to bind. The other thing we also have is that once it's bound, right, that basically will then allow the tRNA to fit into its little active site. So now here's an important piece right here. A lot of times students will be like, well, wait a minute. So how does the tRNA synthetase know which anticodon on this tRNA is the tryptophan one, right? Does it ever select the incorrect tRNA? Right? Is there a phenylalanine tRNA floating around that it could potentially make a mistake on connecting or grabbing? The answer is no. Why? Because of a mechanism for this called induced fit. Activation of the active site. A little bit, right? I mean, because here's how you do this. Here's how you make sure that you don't get the wrong tRNA, because you got tryptophan, right? Now you need to find the tryptophan tRNA. But there's a lot of tRNAs floating around. So how do you make sure you grab the right one? Well, here's how it works. The induced fit idea is actually an important one to understand. And this will probably be the last major concept that I do. And I'll try to start off with this one and kind of move forward from here as well. Here's how it works. Induced fit in this particular case, and I want to talk about this a little bit. So when you take a look at induced fit, what that basically means is when the tryptophan binds to its site, before it binds there, this active site is not an open three-dimensional pocket. That's generally doesn't happen. Usually the way things bind to active sites and enzymes is with induced fit. What does that mean? That means it's like a beanbag chair. If you take a look at a beanbag chair, does a beanbag chair have a predetermined shape? It's a blob, right? It's not shaped like you. Like a normal chair has a predetermined shape. There's a back, there's a seat, and then there's like a little bar for your legs, right? There's a predetermined shape. Now, if you fit that shape, then you can sit down in the chair. So this makes it seem like this open pocket here is a predetermined shape, but it's not. It's like a beanbag chair. What induced fit is basically as a molecule, I create the fit. So the reason why a beanbag chair feels comfortable is because when I sit down in it, I induce my shape on the beanbag chair. The beanbag chair adopts my shape by kind of molding around me, right? So that's my shape. I induce that fit. In this case, the same things happen. So when the tryptophan binds to the synthetase, it changes the shape of the molecule so that its only specific shape change is only appropriate for the tryptophan anticodon. So in a way, the tryptophan molecule induces its own pocket for its own tRNA, so that the tryptophan, in a way, is choosing which of the tRNAs is correct by saying, when I change the shape of this molecule, it's only shaped for tryptophan. the tryptophan tRNA. So that's an induced fit. So that way you don't ever make the mistake of getting the wrong tRNA sliding into this pocket, because the tryptophan has changed its pocket very subtly, but very specifically, so that the only one that fits in there is the tryptophan anticodon tRNA. Okay. So that's induced fit. And then, of course, once you lock it in there, then basically you are ready to just add that to your acceptor end. And that's essentially what happens. So your AMP will leave, and then that tethers and covalently links your tryptophan to your tRNA. And then at that point you release, and now you've got a charged tRNA with a tryptophan. So now it can go to translation and deliver that to the ribosome for incorporation into the protein. Okay. Now then, this is a good place to start. The structure of the ribosome. So Benjamin, from here on out, we're going to be taking a look at the mechanism of translation. And we'll start that up on Tuesday. We'll finish chapter 15 on Tuesday, and the exam on Thursday will be chapter 12, 14, and 15 in its entirety. Okay. Is that a question, Justine?